Hello students, my name is Shashank Shekhar and we are doing the chapter Biomolecule and today is the sixth lecture. So far we have covered the syllabus up till the monosaccharides, starting for the classification. So talking about yesterday's class, we have seen what are monosaccharides. So if you remember, monosaccharides are those carbohydrates which on hydrolysis do not give any further simpler units. Okay, so talking about the example, we have the example of glucose and fructose. So what are glucose and what is fructose? So glucose are carbohydrate which contains 6 carbons and has carbonyls centered at the first position. So for that reason it is also called as aldohexose. Whereas the fructose which has the carbonyl center at the second position, I would say at the second position in proper Fischer projection. Okay, and has 6 carbons, so it is termed as aldoketose. So we have studied about glucose and fructose and monosaccharides. Now if I talk about disaccharides, so what are disaccharides? So disaccharides are those carbohydrates which on hydrolysis give 2 units of carbohydrate, or I would say 2 units of monosaccharide units. So in that we will cover the example of sucrose, lactose and maltose. Moving to higher carbohydrates which contain greater number of monosaccharide units, we have the example of starch, cellulose and glycogen, and they are termed as polysaccharides. Got it? So let us start our today's class. So if I talk about the detailed topics, the first we will see what is disaccharide, then we will see what is sucrose, what is maltose, what is lactose, then what is polysaccharides. So basically polysaccharides is used for the carbohydrate which contain multiple units of monosaccharide units. That can that depend, and the number of monosaccharide unit depend upon the structure of the say plants or animals in which it is present. In polysaccharides we will see the example of starch, and in the starch we have amylose and amylopectin. Then we'll have cellulose. Okay, so let us start our today's class with the first topic, that is disaccharide. So what is disaccharide? So as I mentioned right now, that it is a carbohydrate which release 2 moles of same or different monosaccharides. So both of the monosaccharide units that are produced, they can be same. So you will see the example of say maltose, in which both the units that are produced upon hydrolysis are same. Similarly, you will see the example of sucrose, where the hydrolyzed carbohydrate will give 2 different unit of monosaccharide units. Now we have 2 monosaccharide units, and these 2 monosaccharide units must be joined together to form the disaccharides, and that joint is called, or that bond linkage is called as glycosidic linkage. How it is formed, we will see the mechanism in the next slide. So basically there is a loss of water during this say formation of this bond that is formed between 2 monosaccharide units, and they are linked through oxygen. So there will be oxygen in the middle of these 2 monosaccharide units. So this is about the disaccharide. Now let us start with some examples. First of all, we have the example of sucrose. So sucrose is the most common example, and what do we get upon hydrolysis on sucrose? We get alpha D glucose, okay, and beta D fructose. We have alpha D glucose and beta D fructose. So if I talk about the molecular formula, its molecular formula is C12H22O11. Now if I try to take the common factor in their say moles, then it would give us 2C6H12O6. So if I take this 2 out of here, then you will see that this represent a unit of your say this water, monosaccharide unit, and these are the 2 monosaccharide units, whereas this H2O has been taken out, so it must be having 2 hydrogen more, because 12 to the 24. So that 2 hydrogen and 1 oxygen, that is 6 to the 12, that is now 11, has been taken out in the form of water. Now let us see more about this sucrose. So if you see here, sucrose has 2 units, alpha D glucose and beta D fructose. So these are the 2 units of monosaccharide units, and here we have the linkage between these. So if you remember the previous structure, it was like HOH here. So we have an OH here down the group of the simple alpha D glucose, and for beta D fructose we have OH here. Now upon the formation of this bond, we are left with oxygen only, and OH and H are being removed. That is, your H2 has been removed. Now if you try to imagine the reaction, then it would have gone in this way. That say we have one OH group here, and say we have the another OH group here. Now if I do treat them with acid, or say keep them both in the same mixture and treat it with acid, then suppose one of these is protonated at a time, and now this will attack here and this will leave, leaving us with OH and R and R dash. Now this will be deprotonated again to give us this linkage that is being shown here, and this linkage is termed as glycosidic linkage. Now if you see here, I mentioned earlier that for reducing sugar you must have a free say OH at the first position, but here you can see that the OH has been not placed in the proper orientation, and it is not free. It is say it is attached to this group. You can see here that this is the first position, and there is no free OH, that is the hydroxyl group, on this first position. I would try to generalize that statement. I would not say first position, I would say that position where the attack has been cited. So here you can see that the attack would have been taken place from the 5th of the carbon to the first carbon, and here in fructose the attack has been facilitated from 5th of the carbon to 2nd of the carbon. Okay. So for glucose, the first position should be having the hydroxyl group, free hydroxyl group, and for fructose the second position should have the free hydroxyl group. But you can see that from the structure it is very clear that none of these is having any free hydroxyl group. So for this reason, sugar is a non-reducing sugar. Sucrose is a non-reducing sugar. So how do we define reducing and non-reducing sugar? So we have studied this classification in the first class. So we have seen that if any sugar is say reducing the Tolwyn's or filling solution or our Benedict solution, then we call it as reducing sugar, and if it is not doing so, we call it as non-reducing sugar. One more thing, so how do we define the mechanism here? So if I ask you that how do you define the mechanism, because you have studied earlier in the aldehyde, ketone and carboxyl group that for the say filling solution to react or to get reduced, or the Tolwyn solution to get reduced, you must have a free aldehyde group. Okay, but if you see here, we are talking about the hydroxyl group and not the aldehyde group. Say students, what is happening here is that whenever you see a say chain and a closed structure, there always existed an equilibrium between these two initially. Okay, but since now both of these are locked by the glycosidic linkage, so there is no chance of any kind of equilibrium from going to cyclic to the open chain. Now for that reason, I would say that there is no case of reducing power. Clear? So this was about the sucrose. So I would make two statements here. First of all, that sucrose is made up of alpha D glucose and beta D fructose, and second, it is a non-reducing sugar. And one more important point, that the linkage that is formed between these two units, that's monosaccharide units, is between C1 and C2 carbon. Now how are we saying it C1 and C2? So for that we have to go back to the Fisher projection and see that upon drawing the Fisher projection, means the open chain structure, I would say the cyclic structure also, both of those will show you clearly that which is the first and which is the second. Got it? So we are linking the C1 of the glucose with C2 of the fructose. Clear? So we will call this linkage as C1 C2 C1 C2 alpha linkage, alpha beta linkage. Why I am calling it alpha beta linkage? Because you can see here, sorry, C1 C2 alpha linkage. Why I am calling it alpha linkage? Because you can see here that both of these OH are present downside. On the downside, their stereo is oriented towards the ground. So for that reason, we will call it as C1 C2 alpha linkage. Got it? And why this fructose is say beta D fructose and not alpha D fructose? Because you can see here that the OH is present on the upside. Okay, this OH is present on the upside rather than the downside. So for that reason, this is a beta D fructose. Now let us move to next example. This was the first disaccharide, so sucrose, disaccharide carbohydrate, which is called as sucrose, and it gives 2 units of monosaccharide unit, that is the alpha D glucose and beta D fructose. Now if I talk about the optical properties of this sucrose, then this sucrose is dextrorotatory. Okay, this is dextrorotatory, but on hydrolysis it gives 2 units, first glucose and second fructose. We have seen that, and glucose is dextrorotatory while fructose is levorotatory. Now I would ask you that how could we say predict the rotation of the sucrose based on the rotation for given for monosaccharide unit. So you can predict it by the magnitude of the degree. Now you can see here that fructose has a magnitude of 92.4 degree in the negative direction, whereas the glucose has 52.5 degrees in the positive direction. But the mixture is showing dextrorotatory, that is on the right side. And how do you see it? You can see very clearly that this magnitude is greater. So this will be the determining factor, and this would decide that whether your carbohydrate will go to left or right. So for this reason, we got our sucrose as dextrorotatory. So this is the reason behind it. Now the hydrolysis of sucrose brings about a change in the sign of rotation from dextro to levo, and the product is named as invert sugar. So this is called as invert sugar. So we can put a general statement here. So if you are taking 2 monosaccharide unit and joining them through the glycosidic linkage, then the final optical activity will be defined by that monosaccharide, means whether it will go to dextro and levo. So it will be defined by that monosaccharide which has a greater magnitude of rotation. So we can see here the example of your fructose. Okay, clear? Now let us move to next example, that is maltose. So this is another disaccharide unit. So if you see the monosaccharide unit that are used to make this maltose, then it is same for maltose, means both of the units are same. This is alpha D glucose and this is also alpha D glucose. And what kind of linkage we can see here? We can see a linkage of C1 C4. C1 C4. So we have C1 carbon attached with C4 carbon. Now where is the attacking site of fifth carbon? Means where is the hydroxyl group present at the fifth carbon attacking? In both these cases we have glucose, so it is attacking at the first center. Now if you see for the first unit, we do not have any free OH group for the first unit, that is deploying a C1 carbon for the glycosidic linkage. But the carbon means the glucose that is deploying its C4 carbon for the glycosidic linkage has C1 carbon having the free hydroxyl group. So it has a free hydroxyl group. Clear? Now this free hydroxyl group facilitates the reducing nature of our sugar. So now if you treat our maltose with say filling solution test, or you can say Tolvin's reagent or Benedict solution, then you will get your reduced reagents and oxidized maltose. What is the form of oxidized maltose? It will be carboxylate. Got it? So three things are here to be mentioned. First of all, that maltose is made up of 2 moles of alpha D glucose, means both of the building units, means both the monosaccharide units are same. Second, there is C1C4 linkage, C1C4 glycosidic linkage, that is beta in nature. And third is that they are reducing sugar because we have a free hydroxyl group at the C1 position. Clear? Now let us talk about the next example, that is lactose. So what is lactose? So if you have seen or tasted milk, then you must have seen that there is a kind of sweetness in its taste. We don't naturally, you can say naturally, even though we have not dissolved any sugar, it has a natural sweetness. So the reason behind that sweetness is the presence of lactose. So lactose is present in our milk, and it is a disaccharide which is formed of 2 units, that is beta D galactose and beta D glucose. Now here there is a C1C4 alpha beta linkage because this carbon is having hydroxyl present on the upside, and this has present hydroxyl group present on the downside. So we have C1C4 alpha beta linkage, and it is also a reducing sugar. This one is also a reducing sugar because again we have a free OH group at the first position. We have a free OH group at the first position. Now we have seen 3 types of say disaccharide which has different type of units. So sucrose has alpha glucose and beta D fructose. Maltose has alpha D glucose, 2 units of alpha D glucose only, and lactose have 1 beta D galactose and 1 beta D glucose. Now with this you should also remember the linkage, the type of linkage it has. So sucrose has C1C2 alpha linkage, maltose has C1C4 alpha linkage, whereas lactose has C1C4 alpha beta linkage. Clear? Now this was all about our disaccharide. So these are the 3 examples of disaccharide. You can have more of this, but we have to limit it to only these 3 examples. Now let us move to polysaccharides. So as the name suggests, so polysaccharides are those carbohydrates which releases multiple units, okay, which releases multiple units of monosaccharide units upon hydrolysis, and this is not defined for a given polysaccharide. What do I mean by this? I mean that, see, we have defined that for disaccharide there can only be 2 units, or you can say we can have trisaccharide example, it will have only 3 units. But for polysaccharide we do not have any such compulsions. It depends totally upon the structure of the plant or the animal tissue where that polysaccharide is present. So most of this polysaccharides is derived from our plant and animal tissue. So therefore it will totally depend upon the structure of our polysaccharide, say means protein, the structure of the plant or animal cell, that what would be the length of our polysaccharide unit. It can vary from 100 to 200 say. Now as we have seen that polysaccharides are made up of multiple units of monosaccharide units, and they are again joined by the glycosidic linkage, means the linkage is same for polysaccharides. Now let us start with the first example. The first example is starch. So what is starch? So starch is basically the storage of our plant. They are the main storage polysaccharides of plant, and it is the most important dietary source for human being, because human beings consume plant, so they human beings in a way consume this starch only, and this starch is very important for dietary means for the diet of a human being. Now high content of starch is found in cereals. So if I talk about the plant that contain the starch, then we have the example of cereals, roots, tubers, and some vegetables. And it is a polymer of alpha glucose. What is starch? Starch is a polymer of alpha glucose and consists of two components. So what are those two components? Amylose and amylopectin. We will see what is amylose and amylopectin. They are also polysaccharide. So they are just the two components of starch. So let us study more about starch now. So we have seen what is amylose and amylopectin. They are just a unit of the starch. So if I talk about the distinction between amylose and amylopectin, so first distinction is that amylose is water soluble, means that they can be dissolved in water, whereas the amylopectin it is insoluble in water. Second, thus it is 20% of the starch, means suppose you take starch, then 20% of the starch is made up of amylose, whereas the remaining 80% is made up of amylopectin. If I talk about the structure, then amylo is a straight chain polymer, whereas amylopectin is a branch chain polymer. See, both have the same monosaccharide unit, only the way of arranging those units is different for amylose and amylopectin. So by this we mean that they are linked by different bonding. So if I talk about amylose, so there is alpha 1,4 glycosidic linkage, whereas amylopectin they are joined by 1,6 glycosidic linkage, alpha 1,6 glycosidic linkage. Now the molecular mass varies from 10,000 to 50,000, whereas in amylopectin this ranges from 50,000 to 1 lakh. It ranges from 50,000 to 1 lakh. So as I mentioned earlier, that starch does not have a fixed number of monosaccharide unit. Even I would say that any polysaccharide do not have any fixed number of monosaccharide unit. So therefore the general equation for this hydrolysis is that C6H10O5, that would have n such units, and if you hydrolyze it, acidic hydrolysis H plus, then it would give n moles of alpha D glucose, n moles of alpha D glucose. Next we will move to amylose and amylopectin. So we have two types of starch. Starch is consisted of two types. First is amylose, second was amylopectin. So we have seen the difference that amylose is soluble in water, whereas amylopectin is insoluble in water. Amylose is 20% starch, whereas amylopectin is 80% starch. Now let us look at the structure. We can see very clearly that amylose is linear in nature. We do not have any branching. So what do you mean by branching? So you can see the illustration of branching in amylopectin, where you can see that we have a straight chain here and we have the second chain straight chain here, but we have also some branching. Okay, so it is kind of this. So if I tell about amylose, so we have C1C4 alpha linkage. We have C1C4 alpha linkage. Now I have mentioned earlier that most of the polysaccharides do not give the reducing test. You can see the reason very clearly, that this chain is in long order, and thus both the C1 and C4 are involved in bonding. Okay, C1, it's also C1 is involved bonding. It's also C1 is involved bonding, and none of the C1 has free hydroxyl group. Due to this reason, we do not have any reducing nature for amylose. Similarly, for this amylopectin, the C1 hydroxyl group has been involved in bonding. Next, here you can see that in amylopectin we have C1C4, means there is a linear chain of C1C4, but at the same time we have some linkage between C1 and C6. So here you can see that we have C1 and C6 linkage, and that is also alpha. Okay, that is also alpha linkage. But for this we have a beta linkage, means this C6 that is involved, it is having beta linkage. So it is C1 C6 alpha beta linkage. But this linear chain is having the same kind of linkage as amylose was having. It is C1 C4 alpha linkage. So this was about the structure of amylose and amylopectin, and they both make up the starch. Now let us move to the last type of polysaccharides, that is cellulose. So what is cellulose? So cellulose are made up of n units of beta D glucose. So if I ask you that how is this say cellulose hydrolyzed, then you will give the same equation C6H10O5 C6H11O5 H11O5 with n units, and this upon hydrolysis gives us C12H22O11, and these are present as n units C12H22O11, and this can be reduced to C6H12O6. Okay, clear? Now each of this unit, that is C6H11O5, will be added by this water molecule to make it to this C6H12O6 molecule, and that would be say present in n moles. Now the type of linkage that would be seen here is a C1C4 glycosidic linkage. Sorry for this is written wrongly. We will have OH here rather than OH on the downside. We will have OH on the upside. So this will be formed in this way. There will be 6-member ring, and since it is a unit of beta, so therefore OH will be present on the upside. So therefore H will be present on the downside. H will be present on the downside, and we have already at C4 carbon we have already H present on the downside, upside. So the kind of glycosidic linkage would look like this. We have O here. Again we have O here. So therefore this will just extend in multiple units. So again we have one H here, and one O here. So this will extend up till n units depending upon the structure of say carbohydrate we are dealing with. Now I would suggest you just try to draw the monomeric units, or the glycosidic linkage by yourself, because in exam you may be asked to predict the structure, or you may be asked that which of this is structure of say produced upon hydrolysis of amylopectin. For that you should remember the actual structure. Okay, rest of the say units will be in the same way. There will be no effect on those, so you can, I would leave it like this only. Okay, so this is our cellulose, which is made up of beta D glucose. So let us revise all those say disaccharides and polysaccharide. So far we have seen three types of disaccharide. The first is sucrose, the second is maltose, then lactose. So these are the three examples of disaccharides. And for polysaccharide we have say starch, which is 20% amylose and 80% amylopectin. Whereas the second one is cellulose. If I talk about the reducing nature, then only maltose and lactose out of these say 4 or 5, out of these 5 only maltose and lactose are reducing in nature, because all of the remaining one do not have any free OH group at the first position or at the second position, means on the attacking side. Now if I talk about the units, the monosaccharide unit that makes up the sucrose is alpha D glucose, alpha D glucose and beta D fructose. What is the maltose made up of? It is made up of 2 alpha D glucose. Lactose is made up of beta D glucose and beta D galactose. In amylose, amylose is linear in nature and it is branched in nature. Now both of these have alpha D glucose only. Whereas in cellulose we have beta D glucose. What were the type of linkage that were seen? So in sucrose, if we talk about sucrose, which has alpha D glucose and beta D fructose, we have C1 and C2 alpha linkage. C1 C2 alpha linkage. Whereas in maltose we have C1 C4 alpha linkage. And again for beta D galactose and fructose we have C1 C4 alpha linkage. Now talking about amylose, it has a linear chain which is C1 C4 again C1 C4 alpha linkage. But amylopectin has one more, that is C1 C6 linkage. Means we have two linear chain. Say we have two linear chain of say C1 C4 glucose, and two of such linear chain are attached by C1 C6 position. Okay, C1 C6 position, and we have alpha linkage there. Now if I talk about the cellulose, we have beta D glucose, but the linkage is C1 C4 alpha beta linkage. So if you look at all of these structure, you will be able to define which of these is reducing and which of these is not reducing. This was for carbohydrates. So if I try to just put all the syllabus in one point, then I would say that we have started from carbohydrates. So we have first seen the classification. What we have seen in the classification, that we can have 4 bases for classification. The first one was on the basis of taste. So the one which is sweet in nature, it is called as sugar. The other which is not sweet in nature, it is called as non-sugar. Then the next basis of classification was whether it is reducing or not. So if it reduces our Benedict solution, filling solution and Tolvin's reagent, then it is termed as reducing. If it is not doing so, it is called as non-reducing. The next basis was the number of carbon. So if it has 3 carbons, it is named as triose. If it has 4 carbons, then it is named as tetrose. Then we have pentose and hexose. Then the next basis was the number of monosaccharide unit present in the given carbohydrate. So if it cannot be hydrolyzed further, means that is the basic unit, then it is termed as monosaccharide. If it can be hydrolyzed to give 2 units, then it will be called as disaccharides, or you can say oligosaccharides also. So oligosaccharides are nothing but they are defined for a certain number of monosaccharide unit. For say if we have 2 units of monosaccharide unit to form a single saccharide, then it will be oligosaccharide, or 3 units of 4 units. But it is not defined for polysaccharides. So means polysaccharides are not oligosaccharides, but disaccharides, trisaccharides or tetrasaccharides are oligosaccharides. Clear? So we have seen this classification, and we have seen one more classification that was on the basis of the carbonyl group. So if the carbonyl group was present on the first position, then we can say that it is aldose. If it is present on the second position, then we call it as ketose. So these were the 5 classifications. Then we started with glucose. So in glucose, we have seen that the open chain was not able to open chain structure was not enough to explain all the chemical properties of glucose. For that, we introduced the cyclic structure in which the C5 carbon hydroxyl group attacked the C1 aldehyde group to form a glycosidic bond. I would not say glycosidic bond. I would say a furan form. Clear? Pyrene form. Now this structure was well enough to explain that why these reactions were not seen. So what were those reactions? So if you remember, the first was not the reactivity, means unreactivity toward the Gibbs reagent. The second one was the less reactivity of the pentaacetate. The third evidence was that there existed two forms of glucose. One was alpha and second was beta, and that was not explained by the open chain, but that was explained with the help of the cyclic structure. So upon the attack, if the OH group goes down, then it is termed as alpha, and if it is going upside, then it is called as beta. Clear? So this was about alpha and beta D glucose. Then we have seen some of very interesting phenomena that are seen in glucose. The first one was mutarotation. So what was mutarotation? So if we take any pure form, suppose we take only pure alpha or only pure beta and we dissolve it in water, then after some time we will see that there is a shift of equilibrium toward beta or alpha depending upon the value of equilibrium concentration of both alpha and beta. And at last we will see that the solution will be adjusted in such a way, means the concentration of say alpha and beta D glucose will be adjusted in such a way such that the overall optical rotation of the solution is 52.57 degrees in positive direction. Okay, yeah, one more thing, there will be also the presence of open chain, but that will be present in a small amount. So this was termed as mutarotation, in which the pure alpha or pure beta form in the presence of water changed its concentration to attain the equilibrium. Next one was epimerization. So this was termed as epimerization. Next thing was osazone formation. One more thing, why it was termed as epimerization? So if you remember the definition of epimers, so epimers are those pair of carbohydrate which have same configuration at all the site except one site, and that one site can be any of the sites, say C1, C2, C3, C4. So if two carbohydrates are distinct due to change in the position of OH group on one side, then they are termed as epimer. Like we have the example of glucose and mannose. So glucose and mannose are epimers because at the C1 position of both of this the configuration is changed. Clear? Now next we have seen the osazone formation. What we have seen? The osazone formation. So what is osazone formation? So in osazone formation, what happens? If we treat our carbohydrate with osazone, then it would result in the formation of carbohydrate osazone, and then they can be crystallized easily, which is difficult for the case of sugar. Okay, so they can be crystallized. Now what is the benefit of this? So they can be used for the purification method. So if you have your glucose dissolved in a solution and you want one of those to be purified or isolated, then you can use the technique of osazone to crystallize it and filter it out. Now one problem with it is that two types of carbohydrates can give the same osazone. Like the glucose and the mannose have the same osazone product because they have only different configuration at C1, but the rest of the carbon have the same kind of configuration, means there is a retention of configuration in the later on carbons. Clear? So for this reason I would say that they form the same osazone, and I have shown you some pairs of carbohydrate which can give the same osazone products. So this was about osazone. Then we have seen the structure of fructose, and today we have covered the rest of the disaccharides. Now since we have covered carbohydrates, now in the next class we will move to proteins. So let me give you a brief overview of proteins. So proteins are basically made up of amino acid which are linked to polypeptide bond, and they have the structure defined in a certain orientation, starting from primary, moving on to secondary, tertiary, and then quaternary. They can be denatured, means these structures can be broken with the help of some, say, heating or different chemical methods. So that is a different thing to discuss. So we will discuss this thing in the next class, and also we will discuss the importance of carbohydrate, in which I have not covered it today. I will cover it tomorrow in the next lecture, I would say. So this was for today's class. Thank you.